Bismillahirrahmanirrahim. Bismillahirrahmanirrahim. In the name of Allah, the most merciful and the most beneficent. Countless salutation on the beloved Prophet Muhammad sallallahu alaihi wasallam. How many participants we have? 5. Muhammad Yusuf Jamal, please unmute your mic.
Assalamualaikum sir.
Walaikum salam. Is this your first class?
यस सर सर पहले वाली भी ली है जो अभी पहले एंड हुई है.
Okay. Okay. Mute your mic.
ओके सर.
Jonathan Javed, when we have more than 15 participants, please let me know. We will start. And just wait for
Sure,
1
sir.
minute only. If they will
Okay,
come, it's okay. Otherwise, we will start with the available participants. So,
Okay,
did you
sir.
guys get something from the last lecture? Jonathan Javed.
Yes, sir.
Very good. So, also please everyone just try to use all the utilized tools again. Read about the principles of tools, how they work, in which nature of experiments, and these tools will be efficient. You should know about that if you want to be a good researcher. So last time we just discussed the first 3 chapters of quick guideline for computational drug designing, and we will try to summarize the remaining 4 chapters, 6, 7, 4 chapters in this class, hopefully. How many participants we have? Jonathan.
So we have 6 participants.
Okay, 1 more minute, 30 seconds more for them. Please try to be online at the time of class. Okay? आप लोगों को चाहिए कि कम से कम 5 मिनट पहले, 10 मिनट पहले रेडी रहा करें. सेपरेट रूम जहाँ पे खामोशी हो, आप तवज्जो से समझ सकें. अगर आपसे कुछ पूछा जाए तो पीछे से काफ़ी सारी आवाज़ें बाकियों को डिस्टर्ब ना करें. तो इस तरह का कुछ इन्वायरमेंट ढूंढें. तवज्जो से पढ़ें बेटा. मेडिकल साइंस में सुनने में आया था बहुत तरक्की है, लेकिन ये कोविड-19 ने दिखा दिया है कि मेडिकल साइंस में कितनी तरक्की हो चुकी है. So struggle very hard for yourself, for your country, and for humanity. I think we should start. Bismillahirrahmanirrahim. Okay, who we have with us now? Aisha Tabassum. Please unmute your mic. Aisha
Assalamualaikum sir.
वालेकुम सलाम.
Yes sir.
बेटा स्क्रीन नजर आ रही है आपको?
Yes sir.
Please read the abstract.
Okay sir. Similar sequences may have similar structure. Is the philosophy of sequences prediction techniques. The 3D structure have structural and functional importance of drug designing. Homology modeling, threading, and ab initio approaches are the computational techniques to predict the 3D structures of the proteins for further studies. In this chapter, we will precisely elaborate numerous tools of 3D structure prediction.
Thank you, beta. So this abstract starts with the line, with the statement, and there is also another mistake. I think that time I did not do a good proofread. We missed the apostrophe comma here. Okay, inverted comma, sorry, not apostrophe comma. The proteins have similar sequence. The proteins having similar sequences may have similar structure. It is a very big statement and is the philosophy of sequence prediction technique. How? So we will dissect this sentence, then we will have very good data on that one. The proteins having similar sequences, ऐसी proteins जिनके sequences जिनकी मिलती हैं, similarity रखती हैं, कितनी similarity रखती हैं? More than 70%, more than 65% when we perform blast P, protein blast. Then the blast P just performed to align your target sequence against the available sequences in protein data bank. The structures having the sequence, and then blast P just align the target sequence with the available sequence of the predicted structures. And if the template has more than 65% or 60% similarity, you will have a good result. So the proteins having similar sequence may have similar structure, may have, not always. So that's why when we just perform three-dimensional structure prediction, remember you guys, we first try to find the protein, family of the protein that has the predicted structure. If you are, suppose there is a protein family A and you just have a protein name ABC, but protein family A also has a protein that is DEF and have the predicted structure but through X-ray crystallography or NMR technique. So always use that structure first as a template, because that structure should have maximum number of similarity, the backbone, the similar backbone, the similar physiochemical properties. So try to use the same family protein if the structure is available. This is the very important concept if you want to do structure prediction through computational means. The proteins having similar sequences may have similar structures is the philosophy of sequence prediction techniques. So we will move. Okay, sorry, this what happened. Okay, now it's okay. Again, we have the same statement here. The proteins having similar sequence may have similar structures. This time I have the inverted commas. Then you have structure prediction tools. There are numerous tools available for structure prediction. The story again ends with this conclusion that you guys should know which tool is best for the target sequence you have. And if you don't know about the principles of the softwares or the function or the working of the softwares, it is very hard for you to decide which software you have to use. You should be very clear in your experiments. You should be very keen in your experiments. You should be very focused in your experiments if you want to do the research. And if you just want to finish this course, then you can do anything. But if you want to understand, if you want to absorb it, if you want to make your career in research, if you like research, if you like findings, new findings, experiments, then you should have to be very focused. Firex2. Okay, then we have, this is the, you guys all used it, so you know everything about Firex2. This is the results and how to analyze the domain analysis. This book has all the results of the utilized tools by pictures, by figures, and also the interpretation. So if there are results, there should be some interpretation, some results, some discussion about the tool, that what the result is. And then we also have a conclusion. So it will make you easy to understand about the tool. The main aim of the book was for the layman, for the beginners. Then ITESR is a very good tool by Chung's lab. Okay, we have the reference here, Chung et al. 2010. And this lab have very pretty beautiful softwares in computational biology. You guys should have to use all the softwares, understand all the softwares, their working, their principles, their limitations, their pros and cons. Okay, we have to submit the sequence on ITESR. Then there is the utilized templates, predicted structures, and which structure is structurally closely related to the target protein. Then you have the protein function, ligand binding site. ITESR also predict the ligand binding site with the help of Coach software, COACH. Coach is also from Chung's lab, as I remember. Then if there is a cofactor, like here mentioned in figure number 17, where is figure 17? Yes, if there is a cofactor, like in CYP1 family, or maybe zinc finger family, or any cofactor to any protein, and if you are predicting the structure of that protein that needs a cofactor, it also predicts the cofactor. Then you have RaptorX. Nowadays, RaptorX is also a good software. I observe it. Actually, I'm just moving forward because we studied in these tools in detail in our classroom. You performed experiments on this, and yeah, last lecture we also discussed something about that. Then we have Swiss model. I hope you know the limitation of Swiss model. So nowadays, that's why Swiss model has very less citations. But for the small proteins or the proteins having very good templates, very suitable templates, Swiss model is also a very good software. If you have a template having a similarity of 65% or more than 70%, Swiss model is a very good software in that condition. Okay, if anyone has any question, please raise your hand. Then we have 3D Jigsaw. This is the home page of 3D Jigsaw, and then you have to submit the protein, then you have the results. Like, okay, here we have a loop. You can see this is a loop, big loop. So you can use Modular to remodel this loop for the better results. Then we have the sequence submission in 3D Jigsaw this way. And after that we have IntFold 2. It's also a good software. Every software has their own pros and cons. HHPred, and results output. Then we have M40. How many softwares we have? It's not going to finish. Modular, okay, Modular. Now we have Modular at last. At last, we have the Modular software. Modular is a very good software for homology modeling, in which we have to align the target sequence with the template sequence. Then we have to find suitable templates. You have to calculate, you should know how to calculate the similarity, figures, e-value, everything of the suitable templates. Okay. Then we have, this is the downloading of Modular, and you guys know very well about the Modular. Here we have RASF2 sequence, 326 amino acid. We want one chain A. I already described these files to you in the classroom. This is our alignment file, sequence file, where we just put the sequence. Then we have this BLAST P for suitable template. There are so many templates we have, like we have maximum score, total score, query coverage, e-value, identity, and I think there is not a good template for RAS F2, as the maximum query coverage is 26% and identity is 34%. So in actual, from 326 amino acids, only 26% query coverage. So it is almost 52, 62, 72, 78, somehow 78 amino acid, or maybe 80 amino acid from 326 amino acids. Out of 326 amino acids, only 80 amino acids are present in this first template, and it is the template of RAS protein. And after that we have only 80 amino acids. From that 80 amino acids we only have 34% identity. So it means 34% of 80 will be almost 28 to 27 amino acids. So we have only 27 or 26 amino acids from 326 amino acids in the suitable template. So by this calculation, you can find that the result will not be good by homology modeling. You have to use threading approach or ab initio approach for this one. But we also use this modular software here for the learning. This is the alignment file, align 2D file. The script is available here. Then we have the command mod 14 align 2d.py. Okay, then we have this another model building script of .py Python language, that if you can change ending model 10 to 100, then you have 100 models. This is only for the 10 models, generation of 10 models. And if you want more accuracy, you can change the parameters according to the nature of your experiment. This is our query mod 9.14 get model.py. It's mod 9.14. In the upper slide, upper figure, I just noticed that it is mod mode. Mode will not work. Mod 9.14 will work. Then we have limitation. This portion is very important, interesting portion. I always like this portion. Limitation of structure prediction tools, Firex2, which limitations we have, and then we have ITSR, limitations of ITSR, RaptorX, Swiss model, 3D jigsaw, int fold 2, M40, modular have so many limitations. Okay. Okay, this portion is very interesting. If you guys want to read, I will try to mail you the book. Jonathan Javed, CR, please remind me, so I will mail the book to all of you. Remind me tomorrow morning. And this table is also a very good, interesting table, guideline for structure prediction, in which you will know about the tool name, availability of the tool, description of the tool, workflow, URL, and references. ITSR, RaptorX, Swiss model, 3D jigsaw, int fold 2, HHPRED, M40, modular, and then we have references. This was the chapter number 4. Now we are going to move on chapter number 5. I didn't hear any voice from any one of you, like in class you guys was shouting, sir bas kar dein aur nahi padhna. Now you are in silent mode maybe. Okay, how many participants we have? We have 14 participants. Okay, do we have Hafiz Hanan with us? Abdul Hanan? Okay. Okay, please unmute your mic, Abdul Hanan. वालेकुम सलाम. आपकी स्क्रीन पे शो हो रही है बेटा बुक?
Yes, sir.
Can you please read the abstract for me bit louder?
The computational prediction of protein 3D structure involves the mathematical modeling techniques based on statistical terms. That is why the resultant structures may have some structural issues related to beta sheets, alpha helices, non-structural loops, amino acid chains, angles, and backbone PSI. PSI bonds angle that cause potential energy constraints in the structure. The validation of the computationally predicted structure is mandatory that compares different parameters of the protein 3D structures with that of experimentally available protein structures. Here in that chapter, we will discuss the evaluation prediction of the 3D structure of both their accuracy and physiochemistry by numerous available tools.
Thank you so much. Thank you beta. Thank you so much. So this portion is interesting one. This one I highlight it. The validation of the computationally predicted structure is mandatory. अब ये आगे आंसर भी है क्यों mandatory है. That compares different parameters of the protein 3-dimensional structure with that of experimentally available protein structures. जो experimentally available protein structure है, sorry, where we are. Okay. Sorry, man. Sorry. Okay. Okay. We were here. Okay, we were here. Sorry for that. The validation of the computationally predicted structure is mandatory that compares different parameters of the protein three-dimensional structure with that of experimentally available protein structures, in which we just compare the predicted structure with the available data of the predicted structures with the help of X-ray crystallography and NMR. That an X-ray crystallographic and NMR structure will not be predicted. It is solved structures that has experimental evidences. In computational biology, we usually have hypotheses, opinions, in silico analysis, but in experimental analysis, we have evidences. So we are going down. Then we have motivation, model evaluation tools. Okay, first one is ERAT. It's a very beautiful tool for structure prediction. Oh, sorry, structure evaluation. And this tool helps to analyze the overall quality factor of the protein. ERAT uses the algorithm of protein structure verification that especially suited for evaluating the progress of crystallographic model building and refinement. Brief instructions, you know how to use this one, what will be the input file, how to submit your sequence, and then you have the result like this one. I'm sorry, it's as I already mentioned that this book was written in 2017 and published in 2018. Now it's 2020. Now the ERAT results is amended. Now ERAT showed the results in colors. I remember green, yellow colors. Before that it was like black and white. The lines that is in dark black color. These amino acids considered as not the good amino acid overall quality. But the amino acids that are below the line, this line, considered as the good one. So we have 82.0, sorry, we have 82.075% overall quality factor of the predicted structure of RAS F2. Conclusion, ERAT evaluation tool was utilized for the evaluation of RAS F2 structure and 82.075% of an overall quality factor was observed. Then we have Rampage. It's also a very good tool for the analysis of sci-fi angles and favored region, allowed region, and outliers to find the favored region, allowed region, and the outliers. You guys know how to use this software. And now we are on the results interpretation. Okay, we have result interpretation of ERAT one. Okay, ERAT generated a plot indicated the confidence and the overall quality of the model. A single output plot was given by the ERAT which gives the value of, okay, the results are not much in detail here. You can read any paper that has the results of ERAT. In discussion section, you will find the detailed discussion on the results of ERAT. If you guys are much more interested in the evaluation of ERAT. Then there are number of residues in favored region. What is favored region? Favored region is considered as the amino acids are in their right, are at their right position. Suppose there is a serine 29, so serine 29 should be at that position where it is, then it is considered in the favored region. Usually it is considered that favored region should be 98%. And then we have allowed region. Allowed region means that amino acid number 19 is at position A, but it should be at position B. But if it is at the position A, it's okay for that. After that, we have number of residues in outlier region, and minimum number of amino acids should be in outlier region. Outlier region is considered as the wrong placement of amino acid. So in this case, we have 39 amino acids in outlier out of 326. It should be 1 or 2 or maybe maximum 5. So try to fix the errors. Then we have anolia, in which we usually analyze the z-score and gives non-local normalized energy z-score of the predicted model. This is the brief instruction about Anolia, the homepage of Anolia, and then we have results interpretation. This type of results we will have for Anolia. The summary of the amino acids with the high energy, the total number of amino acids with high energy and its presentation percentage, sorry, the total numbers of atoms, the total numbers of non-local atomic interaction, and all that. Conclusion: Anolia can be helpful to evaluate the three-dimensional structure prediction. It determines the high energy of amino acids. Then we have mole probability. It's also a very good software. I usually use this one and like this one. It's bit tricky to use. Now it was user friendly, but now it's bit tricky to use. Last time I used it, but it has so many good parameters to evaluate the predicted structure. Jonathan Javed, please unmute your mic. Jonathan Javed.
Yes, sir.
How much time we have more? As I remember, we cannot extend our class more than 40 minutes. It is the limitation of Zoom, as I remember. So I don't
Sir.
want to be in the lecture. So we will summarize the topic.
So Yusuf Jamal message to everyone, बस कर दें, ज़्यादा हो गया है.
My question is still exist. How much time we have more?
Sir, we start the class at 8:30 or it's 9 o'clock.
So we have 10 more minutes. So we will try to summarize in 5 minutes. So we have 5-minute question section. Okay, question session. Agree?
Okay. Yes, sir.
Thank you. Mute your mic. Thank you. Okay, we were on mole probability. This is the homepage of the mole probability. Why Yusuf Jamal is saying bas kar de? We are just revising our course. We already did this in our class in detail. He is already late in class. He is late here and now he is shouting bas kar de. Okay, this is the homepage of mole probability. Then we just submit the structure as we did in Irat and Rampage. Okay, Yusuf, we will summarize this one and we will stop, don't worry. Instruction of mole probability sequence submission form. Then we have this result. This is interesting result. Okay, I'm going to zoom for it. Zoom. Okay, now it's better. Yes, protein geometry. Now we have protein geometry. Poor rotamers, we have 54 poor rotamers in this protein. Then we have Ramachandran outliers, that is also 54, 16% almost. Ramachandran favored region, out of 326 amino acids, we have only 210 amino acids. Then we have carbon beta deviation that should be more than 0.25 Armstrong, and 44 amino acids are here. These are this is not the good result. That's why it is in red color. Otherwise it will be in green color if the results will be under the parameters of specific tool. Bad backbone bounds 1 out of 2706, and bad backbone angles 100 out of 3654. So results is not seem much good for this structure. Okay, then sorry, we have to zoom this out. Okay, now we have this result for mole probability, and here we also have something in mole probability just nominate which amino acid is in outlier region, like amino acid numbers, amino acid aspargine, this one in outlier region 0.1% psi angle, and Armstrong is 0.317 Armstrong. Then we have another amino acid threonine, then we have leucine. So it just gives a whole table that describe which amino acid is in outlier region. So we can fix it by Winfoot software and fix the rotamers. I think we should have to summarize our lecture here because time is running out and Yusuf is also in hurry. So conclusion is mole probability showed the poor rotamers, outliers and deviated angles of the predicted structure. Okay, Zoom also told me running out of time. Zoom likes Yusuf actually. Okay, anyone has any question till now? We just summarized 5.5 chapters almost. Anyone has any question? Please raise your hand. No one has any question. Jonathan Javed, please unmute your mic.
Yes, sir.
So no one has any question, so we should have to summarize our lecture here, right? See you in next
Sorry,
lecture.
sir, I didn't hear you.
Okay, I'm saying that we have to summarize our lecture here as we are running of time, and we will continue the other chapters of this book in our next lecture, inshallah, and we'll finish this book. You already perform all these experiments. It is just a revision and to recall what we know. Anyone has any question? Okay, thank you so much for joining and listening patiently.